Okay. So we talked about the actual sarcomere. Now let's talk about the actual contraction. How does something contract? Right? How do muscles contract? And that's by this, the Actin Myosin Crossbridge. So as we mentioned, during contraction, it is only actin which gets moved, not myosin. One way to think about the contraction is maybe think of myosin as a sort of motor, which works to contract the actin filament. So if you want to look at here, the myosin, these little red lines, they actually take these blue lines, the thin filaments, and they actually just push them together like a motor. They just keep pushing them closer and closer together, right? Like a motor would, right? Okay. The way that it does this is in a cycle where one myosin head binds to one actin, and then it pushes it forward towards the midpoint or the midline, and then it unbinds from that actin, and then binds the next actin in line, and then pushes that one, and then binds again, and then pushes that one, binds again, and pushes that one. Essentially, if you want to, like, this is an actin filament, that my hand is an actin filament, and this is a myosin head, takes it, pushes it, takes the next one, pushes it, takes the next one, pushes it, and right? So the difference between actin and myosin, there's many differences. For example, you don't really have to know like that many. They're made up of different components. You don't have to know what the components are, right? Myosin it has these like little bulb heads, whereas actin doesn't. Actin has a myosin binding spot. So the actin itself has a binding site on it where it allows myosin to bind to it, right? But myosin also binds to actin, right? Actin also has, and we'll talk about this later, a tropomyosin binding site. So if you look at the thin filament is these blue balls, right? The tropomyosin has this orange like line that's going through it. It's like a rope or like a vine. It has spots for that, but there's like many differences. Myosin has ATP and ADP binding sites, et cetera, et cetera. There's many differences between them, but yeah. So let's talk about, okay, so the myosin is this red, and it's the one with this like little bulb head, right? It's the myosin head. And the actin is these blue balls like that are arranged in like a rope, right? Yes, it is the myosin that moves the actin, not the other way around. The thick filament moves the thin filament. Okay. So this is like the whole cycle of how it gets moved. Because it's a cycle, you can essentially start at any point, and it makes sense. It should make sense, right? So here it starts at the top and goes like this. Personally, I remember this cycle. I actually like to start this cycle here at the bottom, right? To me, like that seems like a better starting point. And I do recommend watching maybe an animated video. So I don't have one here, but Janelle looked in the chat an animated video to watch after this as well to help it make sense. But I'll just try my best to explain it. So I prefer to start it here on the bottom. Again, it's a cycle. It doesn't matter where you start or where you end. So it starts off like this. So the myosin is not bound to anything right now. Well, it's bound to ATP, right? It has an ATP attached to it. As you may know, muscle contraction, it takes up energy. It takes a lot of energy to contract your muscles, so it requires ATP. This is how it's involved. ATP is bound to the myosin head, but it's not bound to the actin, which is right here, this blue line of balls, right? What happens is the ATP gets hydrolyzed. ATP hydrolysis is a way to get energy from ATP. ATP itself is not a source of energy. The only way to use ATP as energy is when you hydrolyze it or break it down. It gets broken down into two components, ADP and inorganic phosphate, PI, through hydrolysis. What that does is that creates energy for the myosin, and then the myosin then uses that energy to attach to the actin, right? It gets cocked, so it's like in this like inactive state. You give it some energy, it like perks up, and then it has enough energy to actually bind to the actin, as you can see right here, right? It's bound to this actin. one myosin head binds to one actin, so one red ball binds to one blue ball, right? And it still has the ADP and the inorganic phosphate attached to it. Next, what happens is the inorganic phosphate gets released. So this little purple thing comes off, right? What that does is that causes the myosin head from, it's in this position, right? Attached to an actin, it then moves like this, right? And which is called a power stroke. And because it moves like this and it's attached to the actin will also move like this, right? It'll move to the left or to the right, depending on which side of the sarcomere it is. It's moved towards the middle, right? In this picture, it's going to the left, right? So it moves to the left. The actin goes to the left as a result. Then ADP gets released as well. ADP comes off. This makes the myosin actin binding very weak. It's still bound and it's still like won't come off on its own, but it's weakly bound, right? The binding is weak because no ADP. Then what happens is ATP comes in, rebinds to myosin while it's still attached. ATP comes in, it binds, and then it releases and becomes inactive, right? And then the cycle starts again. ATP hydrolyzes, it perks up, and then it binds to the next actin. So you see the myosin itself, because it hasn't moved, and it still goes straight up when it gets like perked up or activated. Because it goes straight up and you moved the actin it was bound to last time to the left, it now will be bound to the actin that's next to it or to the right of it, and it'll push it as well. And then it'll get to the next one, push it as well, get to the next one, push it as well. And there's, on a thick filament, there's many myosins. Like there's a whole row of myosins here that are all simultaneously attaching, pushing, attaching to the next one, pushing, attaching to the next one, pushing, right? Okay. And I'm gonna, now that I explained it, right, okay, somebody asked, when somebody dies, what part of the cycle causes the deceased bodies to be stiff? This is called rigor mortis. We'll actually get to that in a little bit, in a couple of slides, so hold off on that. But now in these next couple of slides, I just want to write down what I explained in words. So assuming we start with myosin bound to ATP and uncharged and unattached to actin, the first step is that ATP will hydrolyze into ADP and inorganic phosphate, which will provide energy to the myosin. Using that energy, myosin head will become charged and attached to the actin. Once attached, the inorganic phosphate will detach from the myosin. This will cause it to form a power stroke, which is the actual moving, right? Moving the actin closer to the M line or contracting the sarcomere. Then once the power stroke is complete, ADP will also detach from the myosin, causing it to be very weakly bound to actin, but still bound. A new ATP will become bound to the myosin head, causing it to detach from the actin and return to its starting position. And then the cycle begins again with the next actin in line. Okay. I will cover that now. Actually, I was going to cover it after the question, but I'll cover it now because I think it might help you with the question. So notice what happens to your levels of ATP or energy once you die. I want to ask that in the chat to anybody who has. What happens to your levels of ATP inside your cells when you die? What do you think happens if you know that? Yeah, exactly. Your levels of ATP would go down, or they would gone because your body's not producing anymore because you're dead. And notice what is ATP needed for in the cycle, right? A new ATP becomes bound to the myosin. So when ATP binds to the myosin, it causes, so it is used for the power stroke as well and to energize, right? But what is it used for first before it can even energize, right? A new ATP is needed to bind to myosin, causing it to detach from the actin and return to its starting position, right? So if you didn't have ATP and you didn't have this step, before it can even do this step and energize it, it has to do this step. ATP has to bind to the myosin to detach from the actin. If you don't, what would happen is you would attach, move it, or like move it towards the center, and then it would just be stuck here. The myosin wouldn't be able to detach from the actin, right? And the sarcomere would forever be shortened, right? Because it would have already pushed it, right? It would have contracted it a bit, right? So if it can't detach, that means the sarcomere can never relax, right? Because you need to attach ATP in order to get the myosin off. If you can't get the myosin off, you can't go back to the actin can't go back to its original position. If it can't go back into its original position, the muscle can never relax, right? So you need ATP to bind to myosin in order to relax it as well, right? You need to let it unbind the myosin head. If there's no ATP, you would forever be stuck in this position, right? And that would cause like the rigidity. Yeah, it's what rigor mortis is this. You'd forever be stuck in this form. Yeah, first ATP detaches myosin and then it energizes. If you can't detach myosin, then you won't energize it either. But so you wouldn't, after you die, you wouldn't continue to like stiffen. So like you wouldn't contract more, but you would always be stuck in like that level of contraction. Well, not forever, because eventually rigor mortis stops. I don't know actually the duration of it. That's something you could probably look up, but it's not needed. Like the main thing they want you to know is how it happens, right? And they may ask you about it, right? That's actually a common question about muscles that they ask. So they say when you have no energy, or when you have a disease that prevents you from doing this, or maybe when you die and you have no more energy, which step of the cycle will you be stuck at, right? And this is one example. So rigor mortis will be stuck at this step. For example, if they say somebody has a disease that prevents hydrolysis of ATP, which step would you be stuck at? You'd be stuck at this step because you wouldn't be able to hydrolyze to move on to this step. If they say there's a problem with inorganic phosphate and you couldn't detach it, you'd be forever stuck at this step before the power stroke, right? That's a common question they may ask you regarding this cycle specifically. Okay. Yeah, and it's actually common that they will ask something like that, right? And actually, I'll ask something similar right now. This is a fairly straightforward, simple question, just testing on whether or not you understood the process, the cycle. So really think back, what does the hydrolysis of ATP bound to myosin directly cause? Okay, please take another 30 seconds or a minute to answer this. While you guys finish up 1 second, I will return in a minute. And on the polling. Okay, so the majority of you picked A, with C being very close behind, so it's fairly even. The correct answer is actually C. I'm glad that you guys at least narrowed down to both of these are the roles of ATP in the cycle. So both A and B are the roles of ATP to release actin from the myosin head from the actin filament and reversible binding of the myosin to the actin filament. However, the binding of ATP causes A, right? Just simply binding to myosin causes A. But the hydrolysis of ATP, which is the breakdown of ATP into ADP and inorganic phosphate, causes C, right? So first, ATP binds, and causes A, release of the myosin head from the actin filament. Then it hydrolyzes, and then it causes binding of the myosin to another actin filament, right? To the next one in line, right? I remember that hydrolysis is of ATP is the breakdown of ATP for energy, right? What do you need energy for? You need energy for the myosin to actually bind to the actin, right? And the reason it's not D, which nobody picked, but just in case I wanted to go over, irreversible binding, because the binding can be reversed by just simply removing the ATP and adding by adding in another ATP. So you can reverse it, and if it was irreversible, then you would have a problem in your body, right? This would be a big issue. So that's why it's not irreversible binding, it's reversible binding. Okay, is that clear? A and C are both roles of ATP in the cycle, but A is a result of binding of ATP, whereas C is a result of hydrolysis of ATP. Okay, any questions before I move on?